Genetics from Yale School of Medicine in New Haven, Connecticut. Over to you, Dr. Lek.
Thank you, Laura, for the kind introduction, and I'd like to thank the organizers for giving me this wonderful opportunity to talk about something that I feel very passionate about, and that's genetic reports and what patients should do if they have a non-definitive genetic diagnosis. And it's something for patients to understand that just because you got a genetic testing, that not all results are actually definitive. So I'm going to go over, as an overview of this presentation, different types of genetic testing you can get, and how you actually go on interpreting genetic reports, particularly in the case where there is a non-definitive genetic diagnosis. Now, understand that I first talk about inheritance patterns and then what is and isn't a definitive genetic diagnosis. How do you interpret that report without a definitive genetic diagnosis, and what to do afterwards, which is also very important and should sometimes be driven by the patient and what they want to actually do afterwards. And I'm going to briefly touch upon one of my research focus, and that's the process of discovering new disease genes. And lastly, end off with free genetic testing programs. I'll talk about 2 examples, but it's not an exhaustive list, but just 2 examples of some of the goals of these genetic testing programs. So first of all, to talk about genetic testing itself. So there are 4 main types of genetic testing, and they and the difference is how much they actually cover. So the first is single gene test. So this is just sequencing one gene of interest, and this is typically done when discoveries are made in a research setting and they need to be validated in a clinical setting. So we're either sequencing just one variant in the gene or the whole gene itself, and this is typically more sensitive because these are guaranteed to have more coverage. And a single gene test can also cover things that are not genetic. So we can look at protein levels, we can look at protein localization in a muscle biopsy, and we can also look at enzymatic activities as a way of doing a single gene test. Another and a very popular way of actually doing genetic testing, a gene panel. So this is a set of genes, typically 100 or 200 genes known to be associated with neuromuscular diseases. And for the focus of this talk, limb-girdle muscular dystrophy. So the idea behind this is that we know that most of the limb-girdle muscular dystrophies are explained away by just a handful of genes. So in terms of the recessive ones, these are your calpain 3, dysferlin, and also FKRP. So the idea is, let's go for the most obvious genes first. And this is a very cost-effective way of actually doing it, and it reduces the burden on the analyst that is actually going through the results. The next is exome sequencing. So exome sequencing is looking at all the genes, all 20,000 genes, and not just a subset. So we're not sort of like, we're not limited to a particular hypothesis that a known gene is actually causing your disease, or that the symptoms you're presenting are the only symptoms, and also the biases of picking that. So there are symptoms that are overlapping with limb-girdle muscular dystrophy that may not be in that gene panel yet, and also novel disease genes. And last is whole genome sequencing, which is doing the whole watermelon. Exome sequencing only covers about 2% of the genome. This is the region of the genome we best know how to interpret. The other 98% and the variants we see from it, we have a much difficulty naturally interpreting, and this is something that is atypical that we would get for a patient, and it's more still in the domain of research. So there are the types of different genetic testing, and a lot of that is empowered by this technology called next generation sequencing, allowing us to sequence lots and lots of DNA fragments simultaneously, and therefore the power of actually being able to look at, say, 20,000 genes at once. However, with next generation sequencing technologies, this uses a short read sequencing, and so there are classes of variants that we can pick up easily and some that we have great difficulty. So the things that most of these genetic tests can pick up are single nucleotide variants. So these are just changes in the DNA letter. So from, say, an A to a T. So it's just a single change. A deletion, this is deleting just small bits, so like a few letters, or insertion of a few letters. So next generation sequencing does a fantastic job of actually detecting these variants. And the great thing about these variants is that they account for most of the mutations that we actually see in limb-girdle muscular dystrophy, But however, it doesn't cover all the mutations we see. There are also tandem duplications, interspersed duplications, inversions and translocations, and also copy number variants. And we consider these more complex types of structural variants. So some of these we can kind of pick up, but we're not very powered to detect it. So there's a high false positive and negative rate, especially with copy number variants also. So this is the pros and cons of each genetic test and what they actually cover. So more specifically, the diseases that they struggle to detect, a lot of these genetic tests are FSHD, which is actually caused by a repeat contraction of the D4Z4 repeats, myotonic dystrophy caused by repeat expansions in genes, in two known genes in the intronic region. So introns are typically not covered in a lot of these genetic tests, only in genomes, and even with short read sequencing technologies, we have a hard time detecting repeat expansions. And the last is spinal muscular atrophy. So the genes underlying the disease mechanisms are highly identical. So short read sequencing technologies have great difficulties in telling the difference between these. So typically with these three tests, people actually order tests that are more sensitive to actually detect these three diseases along with a gene panel and things like that. So sometimes they come with the gene panel and sometimes they come additional that your neurologist will actually ask for. But it's just to say what the limitations of these technologies are and approaches. So next I'm going to go into interpreting the genetic test itself. So to understand the interpretation of genetic tests, we need to first understand the typical inheritance patterns that we see in limb-girdle muscular dystrophy. So one inheritance pattern we see is autosomal dominant. That is where one of your parents is a carrier and is affected by limb-girdle muscular dystrophy. And in dominant cases, you only need one mutation in a disease gene that causes limb-girdle muscular dystrophy, and there's a 50% chance that this will actually be carried by the offspring or the children of this parent. And so these are normally labeled with the new nomenclature LGMD, D for dominant. A subset of these, but something that happens very rarely, is what we call a de novo dominant mutation, and this is when the parents are not carriers and the mutation actually occurs in the egg or the sperm, and therefore the next generation can carry this mutation. And so the mutation again is a single mutation in a gene known to cause dominant limb-girdle muscular dystrophy. Another common, so this is less common, another common form is an autosomal recessive, and that's where your parents are carriers, so they have one defective copy of a gene, but they don't actually have the disease, unlike autosomal dominant, and there is a 25% chance, so 1 in 4 chance, that the children will carry 2 defective copies and then be affected by a recessive form of limb-girdle muscular dystrophy, and these are typically called LGMD, so R1, R2, etc., etc. So knowing the inheritance patterns is important by going when looking at the possible outcomes in a genetic report. So one possible outcome, and this is where patients are happy they've actually gotten the answers to definitive genetic diagnosis. So in the case where a suspected recessive pattern is assumed or thought to be the case in a recessive disease gene, and I've bolded and underlined this for a reason, because it's important that these things actually happen and in the genetic report, that there are 2 variants. We need 2 variants, so both copies need to be defective in a recessive case, and they're also reported as pathogenic and likely pathogenic. So this has to occur to be a definitive genetic diagnosis for limb-girdle muscular dystrophy. And typically what we also look at is we actually confirm that the parents are carriers of the pathogenic variant also. So going back, we want to see that one parent is a carrier of, say, mutation 1, while the other parent is the carrier of mutation 2, and therefore the child receiving 2 defective copies. So in the other scenario of a dominant disease gene, in limb-girdle muscular dystrophy, we're looking for one variant, but however, it still needs to be reported as pathogenic or likely pathogenic in a gene known to cause a dominant form of limb-girdle muscular dystrophy. And typically one parent is a carrier and is also typically affected. So what will typically happen is, if it's not known, that the parent will be re-examined, because sometimes what we see in dominant diseases is two concepts: incomplete penetrance, so sometimes people don't get the disease or get the disease later on in life, or variable severity. So some people are so mild they don't even realize they have the disease. So, and that's why it's important also to actually re-examine. So now that's a case of a definitive genetic diagnosis. So what does a non-definitive genetic diagnosis look like? And it's very important to look at your genetic report and revisit it if you have it. So one is in the case of a recessive disease gene, and this is typically when only one variant is reported as pathogenic or likely pathogenic, while the second variant could be either reported as variant of uncertain significance, and this is covered by another talk, so I won't go into the details, but some examples of ways you can actually resolve and actually provide more evidence is, say, ordering clinical tests such as the MRI and looking at the muscle wasting pattern and whether it's actually typical for your limb-girdle subtype and also gene associated with your limb-girdle muscular dystrophy. The other possibility is the second variant is not reported or detected. And this is what I'm going to call scenario one. And later in the next slide, I'll go into the details of what that actually means. The other possibility, if you have a dominant disease gene, is that the variant is actually reported as a variant of uncertain significance. Again, this will be covered by another talk. The possible ways that clinicians and researchers actually go and resolve and a variant of uncertain significance, hopefully into either a pathogenic or a benign variant. And the last possibility is all variants are reported as variants of uncertain significance, or VUS, or no variants are reported or detected. So this is scenario two, and this happens more common than you think. Just a reminder that only 50% of patients actually get a definitive genetic diagnosis. One thing I'm going to have to stress, because I've talked to a lot of patients showing their genetic report and coming to this conclusion, and I just want to elaborate on this. Multiple variants of uncertain significance reported across different genes does not mean you have multiple muscle diseases. So I'll put this in context. A variant of uncertain significance is a rare variant in the population. And what I mean by that is that not many people in the population has that exact DNA change. But to put that in context, when you screen a lot of genes, and also particularly large genes, which muscle disease genes are, by chance, you're going to start seeing some of these rare variants that are difficult to interpret as pathogenic or benign. And also, just to let people know that even healthy people have rare variants also. It just means you have a DNA change that's not common in the population. And these changes for even healthy people can happen in any gene, and that includes disease-causing genes too. They're called variants of uncertain significance because we're not quite sure if it causes disease or if it's benign. So just to put that in context, that even healthy people, if we did screen them, we would see something like this too. So I'm going to go over scenario one. So scenario one is a recessive disease gene where the second variant is not reported either. It's not reported as pathogenic or it wasn't detected at all. So I'm going to go over possible next steps or alternate conclusions. One is we could do a gene-specific test. So we could do sequencing specifically for that gene, getting higher coverage with the ability to detect different types of variation. Or we can look at supporting gene-specific tests that aren't genetic, looking at protein expression, the localization of the protein, for example. An alternate conclusion could be that you're a carrier of a common disease mutation. So the FKRP founder mutation that I've listed here could be as high as 1% in the Northern European population. So you could just be a carrier of this mutation, and there is no second mutation because there is another gene that's actually causing your disease. Another possibility next step we could do is analyze the data for copy number variants. So typically the data generated by this is not typically analyzed by clinical genetics labs because of the high false positive and false negative rates when looking at copy number variants. So we could either use the gene-specific test or use research methods to actually look at that. We could also reanalyze the data for variants that are typically not interpreted. One such variant is a splice donor position -1 variant. This is not going to mean much to the patients in the audience. This is more for researchers and clinicians to know that sometimes reanalysis can pick up extra variants, and this is what we typically find in our research. You could also request exome genome sequencing. Typically, you would request exome sequencing for cases where you've only done a gene panel to look at the remaining genes also. And so not necessarily look for that missing variant. And the last is, but genome sequencing is typically not ordered. And the last is the enroll and research study, where we actually apply the cutting edge genomic technologies to actually look for either the second variant or another possible gene. So we perform either genome sequencing, RNA sequencing, and the last is long read sequencing to detect difficult to detect variants. And that would encompass some of the variants that I actually mentioned in FSHD, myotonic dystrophy, and also SMA. So long read sequencing can detect that. The only problem with long read sequencing is it's high cost and also still high false positive rates due to low coverage. So it's still in the research realm trying to make that technology better, but it holds that promise. While in scenario 2, where you have no variants reported as pathogenic, one such case that patients and their physician need to revisit is that, is the disease actually even genetic? So we have a patient in this session that will talk about their story in terms of this. Next is, if you only had gene or gene panel sequencing performed, then you can request exome sequencing. So this will remove us away from the bias of, your symptoms and what they could be, to just being hypothesis free and searching for all possible genes, because we know that a lot of diseases have overlapping symptoms. So it allows us to detect either novel disease genes or genes that we would not initially think of that wasn't present in the panel. The last is, we can actually enroll patients in research studies. So this is what I'm highly involved in my day-to-day research, to find possible novel disease genes, but I haven't listed here, and also novel mutations and disease mechanisms in known disease genes. And it's just a reminder, this is an active area of research because we only have a 50% diagnosis rate in rare disease patients, sometimes a little bit higher, sometimes a little bit lower, depending on the disease and the mutations we typically find. So in these research studies, we use new genomic technologies and also analytical methods to help solve the remaining. We can also apply novel analytical methods on new or existing data. So hopefully that sheds light. And the reason why we're able to, achieve success in discovering novel disease genes is we recruit a lot of patients with the same disease symptoms. And so because to associate evidence, we require multiple patients to be sure that the candidate pathogenic variant and novel disease gene is the right one, instead of only having an N of one or one example. And what we do to strengthen that evidence of a discovery of novel disease gene is we normally create a mouse model, or we create another animal model. So for neuromuscular diseases like limb-girdle muscular dystrophy, zebrafish models are also popular given their reduced cost and speed in actually generating them, and their similarities in terms of muscle structure. And so having all that, we could conclude a novel disease gene, which eventually will be added to the gene panel. So that's how research goes about in addressing scenario 2. So just to touch upon people that fit in scenario 2, the best way of actually getting in contact and being part of these research studies would probably be to talk to, first of all, your physician, and then also reach out to researchers that you know are in this field, and also your associated disease foundations. So I'm going to touch upon in the remaining minute 3 genetic programs and 2 examples. The first is the Lantern project, where they perform gene panel sequencing, and this actually replaces the Emory Genetics Lab gene panel, and this is performed by PerkinElmer, where they perform clinical gene panel sequencing of all the genes known to be associated with limb-girdle muscular dystrophy and the ones that are very similar to limb-girdle muscular dystrophy. And this is good for patients that have not performed any genetic testing. And this is a good way of actually picking out people with common causes of limb-girdle muscular dystrophy. However, if you've had some genetic testing, either a panel, gene-based testing, it's unlikely to change the interpretation. Normally, these technologies are already really good. There is a possibility you may detect something new, but normally this technology is really good, and also the interpretation clinical labs perform are very good also, so it's very unlikely to change that. And the last is the Rare Genomes project, where they perform whole genome sequencing. So this is performed by the Broad Institute. It's a research-based genome sequencing, so any findings have to be validated by the single gene testing I mentioned. And so this is good for patients that already had exome sequencing and they haven't had any conclusive diagnosis. And this is unlikely to change the interpretation of variants already reported, like what I mentioned from the Emory genetics. So with that, I'd like to conclude my talk, and and hopefully this is a good refresher of what, say, a genetic counselor went over with you. And it's always good to have a refresher, because when you get that genetic report, it is quite overwhelming, and this is a refresher for you to look back at your genetic report.